Thank you, Ray. Thank you to Precision for inviting me to speak here today. I'm not going to give a data presentation. My presentation is more focused on the development of lipid-based or liposomal nanoparticle formulations. And I just wanted to talk briefly about my presentation outline because I thought it's appropriate to introduce Transferra a little bit more because more recently there have been significant changes. We have been undergoing sort of a name change. We have been undergoing other changes as well. I just want to talk a little bit about this first. And then just in general talk why I use liposomes, introduce the different stages of development. And I think my main conclusion that I want to work towards is that the liposome or liposomal nanoparticle technology is a mature technology and allows to fast-track development. So just by way of background, so Transferra is actually formerly known as Northern Lipids, a privately held company established in 1991. So we are around now for almost 25 years. And we are a manufacturing and development organization since 1996. Before that, it was focused on lipid synthesis. That's something we are not doing anymore. And that's why basically we decided, okay, we need a rebranding. We need to change our name. The focus was on Transferra Nanosciences. So nanosciences actually, because we felt that's the space we're operating in and we should place more emphasis on this. As part of this sort of rebranding, looking for expansion, we of course had conversations with other companies. One of the companies was Evonik, and we actually are getting acquired right now by Evonik. This is part of their expansion into the nanomedicine area that Evonik has identified as a future, sort of area of interest and focus, and they have made already significant investments into this area. So having said that, sort of the technology our technology focus is sort of complex nanoparticle-based formulations, in particular liposomal or lipid-based formulations, but we have also worked on polymer, inorganic nanoparticles, and to some extent, some conventional formulation as well. Two facilities, the R&D facility, which I am heading, and we have a clinical manufacturing facility that is capable of doing everything from phase 1 to phase 3. Staff is about 50. So with that, I want to go into sort of my actual presentation. Liposome technology is a mature science. Right now, we have around 13 liposomal drug products on the market, over 60 or even more in clinical trials. A lot of clinical trials actually also sort of being initiated in the area of nucleic acid delivery. I'll come back to this a little bit later. Why use liposomes? One of the main uses is of course, just as a solvent to improve the solubility of hydrophobic drugs. This does not really take advantage of the delivery sort of aspect of a nanoparticle. Because typically sort of if you incorporate a hydrophobic drug into a liposome bilayer, it's not retained very well. So it really acts more as a solvent for this drug, and upon following administration in vivo, it's actually rapidly released. So it's basically the main aspect here is solubility and safety, the use of safe excipients. One of the main uses is, of course, to change the PK and biodistribution of specific drugs, and that's if you really manage to stably incorporate a drug into a liposome, and the design principles to design long circulating liposomes, they are very well established. You can actually target specific organs, mainly organs where you have discontinuities in the vasculature. This could be tumor sites of inflammation. And that allows basically to alter the accumulation, in these organs. Another aspect is, of course, to provide protection against degradation. Some drugs, they are simply not stable at neutral sort of pH conditions. For example, one is topotecan. The incorporation in a liposome that has a low pH sort of stabilizes this drug. And in particular, in the nucleic acid field, RNA and DNA RNA-based oligonucleotides, or mRNA, they can be stabilized against degradation by using sort of lipid-based carriers. Overall, it results in these changes, or this results in increased safety. The drug is sequestered away from organs, that are sensitive to this particular drug. I have mentioned doxil. Efficacy, is one aspect, increased drug concentration at disease sites. Makebo, whether this is really true or not, but I was part of the development of Makebo, so that's why I put it there. The picture just shows basically on top. The top picture is basically just it shows the accumulation basically of a fluorescently labeled sort of formulation in a tumor. So this is the lower panel, whereas the top is just that there is no accumulation just based on normal vasculature. So the liposomes cannot really escape at that point. So why use liposomes? They also make us sexy. So there's a lot of activity going on in cosmetics. So the point I want to make with the next slide, it is a versatile formulation platform. I want to start basically on the left-hand side and just go in a clockwise fashion. So I start with proteins and peptides. You can basically associate proteins and peptides, for example, with the external surface. This could be based on electrostatic interaction, could be any other interaction as well. Next, if you move ahead in a clockwise fashion. So I have just an example, sort of a lipopeptide, but that could be any lipid-modified drug, basically a prodrug, because very often, you can take an approach where you modify your prodrug with a lipid anchor to incorporate it into the bilayer. That would be more a MEDCAM approach to formulation, because you modify the drug specifically so that you can formulate it in, for example, a lipid carrier. Others, there are naturally occurring lipopeptides. There are other, sort of lipids, for example, monophospholipid A, that are actually lipids, and are used as adjuvants in vaccine formulations. Then if you go down to verteporfin is actually a hydrophobic drug. It's incorporated in the bilayer. As I said, it's rapidly released after administration, so it's not really retained, and that's the fate of most of the hydrophobic drugs that you incorporate, unless they're really lipids or very lipid-like. At the bottom, of course, I had to put down doxorubicin, which can be accumulated based on its weak base properties by using a pH gradient loading sort of technique. You could also use a complexation technique because it forms complexes with metal ions. So they are different, and these are so-called different remote or active loading techniques that allow you to accumulate sort of the drug in the liposome interior. Most often, or in the majority of cases, the drug would actually precipitate out because the concentrations are so high that the drug tends to precipitate. And that allows basically to really retain the drug inside, and you can achieve sort of this accumulation benefits at disease sites. Of course, the last one is the nucleic acid delivery. This could be RNA, DNA oligonucleotides, messenger RNA. And that has really taken off since the development of more and more powerful cationic that actually also are important basically for intracellular delivery, and is basically activity of the mRNA. With that, sort of the difference, the definition what the liposome is has become much more sort of expanded, because on the left-hand side you just see a cryo-electron picture of a liposome. In the center is basically a cryo-electron microscopy picture of an oligonucleotide formulation with a liposome. So you see these concentric multilamellar structures. On the right-hand side, basically so what we would call now a solid lipid nanoparticle. So, but the difference between these is just very often it's just lipid ratio, for example, between these two in the center. So where you, at the one hand, get something that is multilamellar. All of these are below 100 nanometer. On the other hand, you get something that is really a solid lipid nanoparticle. So liposome manufacturing strategies, they are sort of very well defined. So I just want to go sort of briefly through a conventional, sort of the conventional strategy, typically comprised of 4 steps. The liposome formation, the size reduction step, purification, concentration, and stereofiltration. What we, from our aspect as a CMO, we are very often faced that people bring in sort of a process that was based on thin film hydration, which is not really a very scalable process. So we always try to get immediately away from that. Solvent dilution or solvent injection is a much more scalable process. Sometimes it will be sort of formulating a hydrophobic drug in a lipid powder and basically doing lyophilization and then hydration of the lipid powder. Size reduction, of course, from our aspect, we do build commercial extruders, so extrusion is one technology that we commonly use. I had to show one picture, I promised just to show one, a tiny picture. But it is sort of commercially the most advanced sort of technique right now. Purification or concentration, tangential flow filtration. So I'm showing here a larger scale system that we have built to handle basically larger volumes in the hundreds of liters. And then stereofiltration. What I want to point out too is in particular the last 2 aspects, tangential flow filtration and the stereofiltration. That's also sort of processes you have to really pay attention to. There can be changes on, for example, tangential flow filtration due sort of the shear rates that particles experience there, and so it requires sort of some optimization. With that, I want to sort of also emphasize the extrusion-free processes. On the one hand, which basically, if we go back, the extrusion-free process is combined these 2 steps into one. So the liposome formation happens actually at the point where you do the mixing, and the size is actually determined at this point. The downstream processes are again the same processes as we had before. So I want to point out sort of the microfluidic mixing, of course, Precision NanoSystems, their nanoassembler scale-up system, which is right now installed in our facility and basically for cGMP manufacturing. So we have worked sort of over the last months with precision on doing that. There are other approaches, of course, T and Y impingement stream mixing that are very often just custom-based systems. Solvent injection, is another one. I just want to briefly touch on the different stages of the development of a liposomal formulation. The R&D program, which is, of course, focused on identifying a lead formulation based on, for large-scale preclinical testing, based on both pharmaceutical and pharmacological attributes. It involves that we prepare at that point small-scale batches and characterize these batches, so prototypes. We identify at that point, or I should say we actually only choose at that point, sort of processes we are confident they're scalable, develop the basic analytical methods, establish basic storage conditions, so this would be preliminary stability studies, and then, of course, screen prototypes in vivo as well. That's not something we are doing, but we are providing the formulations to do that. Preclinical program really is focused on developing the scale-up manufacturing process and basically where you scale up to the batch size that you would be using for toxicology and clinical studies. This is also the point where you characterize your analytical methods further and qualify them. You establish in-process tests, develop drug product specification, and then, of course, manufacture the test articles. We call the batches test articles for toxicology studies, and perform sort of stability analysis, and these are long-term stability studies. So this is just an example of drug product specifications. If anyone is interested, can talk to me, afterwards. Scale-up process development, at that point, we really focus on preparing intermediate, scaling up via intermediate and large-scale batches. Intermediate would be in the hundreds of milliliter, large scale, would be reflective of the scale that you'll be using for toxicology and clinical testing. And then it's important to really characterize every process step, and that's why I mentioned it's not only the formation process, but it's also the subsequent, the downstream processes like tangential flow filtration and stereofiltration. So development timeline, I put something up there that's a common timeline. It's a little bit of an oversimplification. So I assigned a time to these different stages. Typically, we see sort of 11 to 15 months. And of course, the oversimplification here is, stability testing and how much stability data are already available, and what is the comfort level, of somebody having less stability data available. And very often, that's where we are pushed, to proceed with sometimes not sufficient stability data. And that's, of course, a risk, and some clients are willing to do that. So in summary, the liposome technology is a mature technology. Many of the developmental and regulatory challenges, they have been observed over the past. This allows actually for rapid development, so you can go very rapidly from a prototype to clinical manufacturing. I put up here 1 year, and of course there's a range. And important is really the systematic investigation at each stage of drug development, so that you can actually fast-track these technologies. And with that, I am basically done, and would like to thank Precision again for inviting me.
So we'll take any questions at this point for Norbert? Thanks, Norbert. That was a great overview. Can you maybe just comment on, you've got your stages where you, particularly maybe the downstream stages, because we certainly get a lot of questions on that. Maybe talk about a bit more about what you've found with your experience in tangential flow. What's the critical thing?
I think the critical thing here really is, because it is something that produces shear, So, and of course, shear rate is something that we will typically investigate. What is the effect of shear rate? Do you trigger any particle size changes? Some formulations even, you see at that point, if the shear rate is too high, it's not only changes in particle size. Sometimes the lipid composition can change as well. Shear is anything. It's basically at the diafiltration step, I mean, if you have particle size growth, you're operating with already with 100 nanometer particles, you're getting very close to where it becomes difficult afterwards, to stereofilter. So I think it's really, you have to really characterize all these steps, to ensure that your final step, the stereofiltration, actually works out, And I mean, of course, if you want to avoid, changes or surprises at the filtration or tangential flow filtration step, then you have to characterize it carefully.
So around what you're seeing in your experience in the stability of these lipid nanoparticles, liposomes, an idea for sort of where you think things are at a bit, and maybe I hear a bit about people's success or failure with freeze drying and to affect better stability. But I guess just your opinion briefly on where you think improved stability around to maybe get out of cold chain and things like that might be for the future of the...
I mean, stability is really just a generic term, because there are so many flavors of liposomes, And I mean, just thinking of liposomes that are similar to Toxil, they are actually liquid formulations. They have a storage shelf life of at least 18 months, that's Toxil. And I think very often, I mean, the liquid formulation, so you do achieve, appropriate storage shelf life, but that depends on the use of appropriate liquids as well. So having said that, sort of the lipid nanoparticle system in conjunction with RNA or DNA-based formulations is, of course, one thing, that are just entering the clinic, and there's relatively little known, so sometimes, most often people would do preventative. I mean, you already know, sort of room temperature storage is maybe not the best storage temperature. So it's preventative freezing, Is it minus 20? Is it minus 80? So freezing is, of course, one aspect, and many people tend to go ahead, if freezing is a possibility, tend to go ahead with freezing. The other option is, of course, lyophilization, but the lyophilization is, again, it's a costly sort of process, a lot of development work, and whenever you make changes, you have to redevelop your cycle. So it's not something that we do typically recommend at this stage, but it is certainly an option, to that as well, So, but what it requires in the end, is if you look at your drug product, it requires aseptic lyophilization. So in the end, you have to have aseptic lyophilization.